All right, welcome to The Explainer. Today, we're diving into a pretty radical idea, one that might completely flip how you think about your own immune system. We all know it's a defense shield, right? But what if it's something way, way smarter than that? The standard view is that our immune system is a fortress. It's there to fight off invaders one by one. But this new idea suggests something so much bigger. What if our bodies are actively, deliberately engineering weaker viruses, not just to protect us, but to protect the entire human species? So where did this wild idea come from? It's from a Brazilian bioinformatician, Michel E. Beleza Yamagishi. Back in 2021, he published this paper suggesting our immune system has this hidden second mission. It's a hypothesis that totally reframes how we see viruses, not as a simple war, but more like a really complex evolutionary dance. Okay, a vaccine factory inside our own bodies? Sounds a little like science fiction, doesn't it? So how could that even work? Well, the secret apparently lies with this tiny, specialized army of enzymes that are already inside our cells, fighting on the front lines.
The core process here has a fancy name: virus sequence hypermutation. But honestly, let's just think of it as molecular editing. It's like this. A virus gets into one of your cells to make copies. But before it can, these little editors, our own enzymes, jump in and start making changes, literally rewriting the virus's genetic code.
So who are these editors? Well, the main ones belong to two enzyme families, ADAR and ApoBec. Now, we've known for a while that the ApoBec family is a total powerhouse against viruses like HIV. But here's the really cool part. They leave behind a very specific clue, a calling card. They change the genetic letter C to a U. That C to U swap is the key footprint we're looking for. So let's walk through the whole assembly line. Step 1, a virus gets into a cell. Step 2, as it tries to make copies, our ApoBec enzymes start scribbling edits all over its genetic blueprint. Now, sometimes those edits just destroy the virus completely. But other times, and this is the key, they just weaken it. They create what's called an attenuated version. And this milder but still infectious virus gets out and starts spreading, basically acting as a natural vaccine for everybody else. It's a fascinating theory for sure, but is there any actual proof? This is where the story gets really compelling, because the hunt for these molecular footprints kicked off right in the middle of the COVID-19 pandemic. And the timeline here is just striking. As early as March 2020, scientists were already finding those exact CDU mutations we talked about in the SARS-CoV-2 genome. Then, in August 2021, Yamagishi puts out his paper connecting this evidence to the whole vaccine factory idea. And then think about what happened next. Variants like Omicron showed up. Highly contagious, but much less severe. It seemed to fit the pattern perfectly. And it wasn't just Yamagishi seeing this. Another scientist, Dr. P. Simmons, was looking at the same data back in 2020 and saw this huge flood of CDU mutations. He called it evidence for a potent host-driven antiviral editing mechanism. What's so important here is that you have multiple independent scientists all seeing the same strange fingerprints in the data. Of course, an idea this bold is going to get some pushback, and the skeptics raised some really good points. For this whole vaccine factory idea to be credible, it's got to have some solid answers. So the first big objection really came down to the numbers. If you took any two samples of the virus, on average, they only differed by about 9.6 genetic letters. These are called SNPs. Now, out of a genome with 30,000 letters, 9.6 is almost nothing. The virus looked incredibly stable, which leads to a really powerful question, right? If the virus is so stable, with the mutation rate that low, how could this editing process inside our cells possibly be significant enough to create weaker strains and actually drive evolution on a global scale? And the counter-argument is, well, it's stunningly simple. Turns out, you don't need a whole bunch of changes. Research shows that sometimes all you need is one. A single, strategically placed mutation can be enough to completely change how a virus behaves. And that brings us to the second big pushback. Most of these edits are silent, meaning they don't even change the proteins the virus builds. So skeptics asked, how can they possibly help? Well, this is where the theory gets really clever. A silent mutation is actually the perfect tool for a natural vaccine. Why? Because it can weaken the virus by, say, slowing down how fast it copies itself, and here's the genius part. So let's step back. If this hypothesis holds up, what does it actually mean for all of us? The implications are enormous. It could give us a whole new playbook for how we fight pandemics in the future.
So why does this matter? Well, think about it. If our bodies are already doing this, maybe we can develop therapies to just boost that natural ability and end outbreaks faster. We could copy nature's strategy to design better, cheaper vaccines. And fundamentally, it would totally reframe our relationship with pathogens. We wouldn't just be victims anymore. We'd be active players in our own coevolution.
Now, it's really important to say this is still a cutting-edge idea. It's a hypothesis. The source material calls it a hidden gem that's still waiting for more research. It's really a call to action for scientists everywhere, virologists, immunologists, to go and put this theory to the test. Which leaves us with one final powerful question to chew on. If nature really has equipped us with this incredible built-in vaccine factory, why aren't we trying to optimize it? The next great challenge might not just be understanding this hidden power, but actually learning how to harness it. Thanks for joining me on The Explainer.